All right, so here we are talking about transport across a membrane. So the diffusion of a substance between two sides of a membrane thermodynamically resembles a chemical equation. A, our molecule that we're moving from a source to a destination, can also go backwards, destination to the source. It's all perspective. The difference in the concentration of a substance on two sides of a membrane generates a chemical potential difference. Our chemical potential can be measured as a delta G equals RT times the natural log of concentration of our substance in the destination divided by the concentration of our substance in our source. So effectively our products and our reactants, much like this right here is our, let me get my pen, products, reactants. Now, the movement of ions across a membrane also results in a charge difference generating an electrical potential difference. Okay, so if something is positively charged and it moves to the opposite side of the membrane, that's moving a positive charge across. And so we account for that by looking at the electrical potential, which is Z sub A times Faraday's constant times delta psi. So Z sub A is our charge of our ion, F is Faraday's constant, and delta psi is our membrane potential. Our units for this are in volts. Now, what we are able to do is combine those two things to make our electrochemical potential difference. So this accounts for the species that's being moved based on its concentration with our first part here, and then what's the charge difference of that. Now, thinking ahead, if we're talking about moving something like glucose, well, does glucose have a charge? Is glucose an ion? No, it's not. And so it doesn't have a charge, so we don't have to worry about that part of the equation. But if we're talking about moving like sodium ions, then we absolutely do. Now, membrane potential. A from the source to the destination. Let me get my eraser again. Is this? Okay, there we go. Electrochemical potential difference calculation. There we go. By default, delta psi is measured as the potential inside the cell, the destination, minus the potential outside the cell, the source. A negative number, so this is the convention, whenever you see a delta psi measurement, it's measured as the potential inside minus the outside potential. A negative number indicates the inside of the cell is more negative than the outside of the cell. If an ion moves from the outside to the inside, then the membrane potential is fine. So you don't have to worry about changing a sign or anything like that, because this is our convention where we're measuring something inside minus outside. Okay. However, if an ion moves from the inside of the cell to the outside, so if it's being exported, it's being released or trafficked outward, then the sign of the potential must change. Almost always, the cytosol is considered the inside of the cell, and that's how we're going to operate. We're going to default to this is the cytosol is inside. Now, if more than one ion is transported, you need to calculate the additional energy charge, calculate the free energy for one ion, then multiply for additional ions. Okay, so then the first question is it favorable from a charge standpoint to transport a chloride ion into a cell with a negative 60 millivolts? And what about transporting it out? Okay, so this is a rough idea of how would you calculate this, but let's go ahead and move on to the next slide where we can actually calculate it. Now, transporting sodium ions in and out of a cell with a delta psi of negative 60 millivolts. Our cytosolic Na is 12 millimolar, and our extracellular Na is 145. Now, our equation here is delta G, which, remember, if we have a negative delta G, it's a favorable reaction, is equal to RT times the natural log of the concentration and our destination, AD, over our A source. Now, our source, it's always important to remember that we are looking at this as, now our source is going to be the extracellular concentration of 145, and our destination is the cytosol of 12 millimolar. Now, what we also have to consider with this calculation is the electrical potential difference. And so that would be our Z sub A F times delta psi. Here's our delta psi. Z sub A is going to be, well, it's a sodium ion, so that's plus 1. And then Faraday's constant is 96,485. Okay, so let's go ahead and set up the first part of this calculation. Delta G is equal to RT, natural log. Now I'm going to put my concentrations in. The natural log of 12 over 145. Okay. Next up is RT. Well, what is my R value? That's going to be the 0.008135. What's my temperature? Well, this is at 37 degrees Celsius. And 37 plus 273 is 310 times the natural log of 12 over 145. So that's everything that we need to do for that specific calculation. Now, as we plug these numbers in, we're going to see that we're going to see that work already done right here. So I'm going to erase. Okay, and I've got my R, my T, natural log of 12 over 145, amounts to negative 6.42 kilojoules per mole. Okay, so that's a favorable reaction when we're sending sodium into the cell based on only the concentration, because I haven't done this part of my calculation. Now, you can round those numbers and come up with, this is basically that same thing, but a quicker calculation. Effectively, 0.008315 times 310 is more or less 6. Then rather than taking the natural log, if you did the log of 10 over 150, that'd be 6 times the negative log, or 6 times log of 10 to the negative 1, which will ultimately be negative 6. Now, I want you to do this one right up top, because then you can plug it into your calculator and get real numbers. Now, the other part of our calculation, we've got our negative 60 millivolts and our delta psi, which is, delta psi is listed typically in kilojoules, so we've got to figure out a way to kind of reconcile our values. It's kilojoules per volt. So what we've got is a positive 1 is our Z sub alpha, times 96.485 kilojoules per volt, and then negative 0.06, which is the same as 60 millivolts. We're using volts as opposed to millivolts for this calculation. You plug that in, and you end up negative 5.79. Okay, so what we end up having is, well, a delta G for this of negative 12.21. Great. Now, I like this calculation. I think it's valuable because this gives us a kind of foundational calculation. Like, what could we change about this, and how would it change our answer? For starters, we were using a sodium ion, so that influenced this number right here. What if we used a chloride ion? How would that impact our result? Well, it wouldn't be negative 5.79, it would be positive 5.79. Okay, so the electrical potential would be unfavorable. But then if we think about this right up here, if the concentrations did not change, well then this would not change, because remember, the chemical potential is simply measuring, is this a favorable reaction from the perspective of the concentrations? Whereas our electrical potential is saying, all right, well, what about our charges? So I think that it's helpful to ask yourself those questions, like, how could I make this, how can I make this unfavorable? Well, one way that you can make this unfavorable is if, as opposed to going into the cell, we're going out of the cell. What would that mean for this value right here? Rather than 12 over 145, you do 145 over 12, in which case that's probably going to make this a positive value. Now, if we were talking also about a negatively charged ion, it's going to make this negatively charged, which would make that positively charged, which would make our delta psi positively charged also, because if we're going out of the cell, then our delta psi is not going, we got to switch the sign of our delta psi. Okay, so I think this is helpful for all of those reasons. Now, on the next slide, there's another example problem. I would study this slide, study this calculation, and then account for, well, what if this was different? Would it be favorable or less favorable based on different circumstances? Okay, so this is the same thing, but we're flipping it. We're going out of the cell, and here's that calculation. The chemical potential becomes positive. The electrical potential also becomes positive because we switched our delta psi. We switched our delta psi, and we switched our value of this right here. All right. There you go. There you go. Now, this goes on to say, well, this was the rate for one molecule. What if we're transporting 2 sodium out of the cell? Well, then we double it. Okay, now on to transport. So that was where we were talking about moving an ion. So when it comes to transport, molecules can be transported in a mediated or a non-mediated fashion. So first of all, there's non-mediated transport, also known as simple diffusion. The rate of diffusion depends on the concentration gradient and the substance's solubility in the membrane's nonpolar core. So as we move through that lipid bilayer, is it going to face resistance because of it being polar as opposed to nonpolar? Water, O2, and steroids can diffuse readily across a lipid bilayer. And our model for that is this right here. We're going from a high concentration to a low concentration, and it's a small molecule moving without any sort of protein. Mediated transport. 2 categories depending on the thermodynamics of the system. There's passive mediated transport or facilitated diffusion. Molecules flow from high concentrations to low concentrations. And then there's active transport. A molecule is transported against its concentration gradient. This endergonic process must be coupled with an exergonic process to make it thermodynamically favorable. So that would be our last one over here, this guy right here, where we're moving these 2 things from a low concentration to a high concentration. In order for that to happen, we've got to hydrolyze ATP so that they can. Our passive mediated transport or facilitated diffusion, well, we've got these things that diffuse through a lipid bilayer. They're moving from high concentration to low concentration. Facilitated diffusion, well, we're moving from high concentration to low concentration, but there's a protein involved in that as well. Some examples of this would be something like ATP synthase is a protein that synthesizes ATP. It also moves protons from a high concentration to a low concentration. For both simple and facilitated diffusion, solutes only move in the thermodynamically favored direction. But proteins may facilitate transport, increasing their rates of diffusion. And there are some important distinguishing features. Facilitated requires assistance generally from a protein. Transport displays saturation kinetics. And once saturated, the only way to increase transport is to increase the number of transport proteins. This is a lot like enzyme kinetics. Now, we see that here where we've got proteins that facilitate transport. They can increase the rate of transport. Solutes can either go along their concentration gradient through facilitated diffusion or against their concentration gradient in active transport. Now, our transport proteins that we should be familiar with, or that I want you to be familiar with, there's basically 3 different ones. There are Uniports, symports, and antiports. A uniport moves one substance across a membrane. A symport moves two in the same or the sim direction. An antiporter moves two in opposite directions. So they are opposite, anti. There are also channels or carrier transporters. Channel transporters have a pore through which the solute passes at a high flux rate, or at a very high rate. They show high selectivity, rapid flux rate, and tend to be gated. So what that means is they have control. They have something that gates them and then something that opens that gate. Examples are porins, gap junctions, and ion channels. We'll talk about a couple of these in a little while. Carrier transporters bind to the solutes on one side of a membrane and undergo a structural change, releasing the solute on the other side. These are divided into pumps and transporters. We'll also talk about a carrier transporter in just a little while. Now this, yeah, there you go. There are substances that are transport inhibitors. Transporters can make excellent drug targets because they move a substance from one side of a membrane to the other side of a membrane. So, for instance, what we have up here is the pathway by which tryptophan is converted to serotonin. Serotonin gets packaged up into these nice little vesicles, and then those vesicles fuse with their membrane and release serotonin, and that serotonin is released from one neurotransmitter, or from one neuron, I'm sorry, to another neuron. Now what's going to happen in this other neuron down here is that this other neuron is going to be stimulated by serotonin and cause, some sort of downstream mechanism, some sort of downstream pathway to be initiated, but then it's going to release them. And then serotonin will be taken back up by a transporter. And this transporter protein will put the serotonin back into a vesicle, and ultimately it can find its way going back. Well, there are drugs known as SSRIs, or selective serotonin reuptake inhibitors, and they block this right here. Those transport inhibitors are, those transport inhibitors are designed to modulate mood and impact, like antidepressants are selective or SSRIs are an example of an antidepressant. Transport inhibitors can also cause disease or death. Carboxytrachloside and bongcric acid are inhibitors of the mitochondrial ADP-ATP translocation, so they can stop ADP and ATP from getting in the right location, so they're ultimately going to be deadly. Now this class of proteins I think are fantastic. These are the porins. Now if you look down on them, we've got a top angle view of them. These porins, what they do is they basically enable the movement of a molecule from one side of a membrane to another. But it's a series of beta barrels. So secondary structures of beta sheets that kind of aggregate together to form what's known as a beta barrel. These porins are the simplest membrane transporter. Sorry. They are located in the outer membrane of bacteria, mitochondria, and chloroplasts. The structure of a porin determines its selectivity, and these are always open, and the solute can travel in either direction. Or when they are open, the solute can travel in either direction. Aquaporins. These I think are really just fascinating because I think that it was always intriguing to see a connection between the anytime I see something that's single molecule, it's really, really pretty cool to me. So aquaporins are what you would imagine. They are water-specific pores. Now they allow water to transport at a very high rate. This is worth noting because if you remember, water is a substance that can transport across a membrane kind of freely. It can diffuse across a membrane. Now, aquaporins are very abundant. So this is just a list of aquaporins and different cell types and organisms that they're found in. Yeast plasma membrane aquaporin, plant plasma membrane, liver, kidney, all these aquaporins do different things. Now, when we look at the structure of an aquaporin, what it is, if we look over here, it's a series of alpha helices. Now these alpha helices kind of organize to make a very small pore. Now, I like to look at this model in the upper left-hand corner, because what we've got here is an extracellular side. We have 1, 2, 3 water molecules. And those 3 water molecules are basically being funneled down. This funnel is 3 water molecules wide, and then it's 2 water molecules wide, and then it's 1 water molecule wide. And these aquaporins are going to let a single water molecule cross the membrane at a time. So we're moving a single water molecule from one side of the membrane to the next. Now that's pretty cool to me, because whenever I go back to like Gen Chem 1, and we talk about a mole, you're talking about a huge number of a substance. This, we're talking about just a single water molecule. Not only we're talking about a single water molecule being moved across at a specific rate, but it's actually being moved in a specific fashion. Because if you look, what's highlighted here are a couple of different amino acids. We've got an amino acid that's responsible for some electrostatic repulsion, some size restriction, and then water dipole reorientation. Effectively, what that means is that if a water molecule rolls up like this, what this aquaporin is going to do is it's going to say, "Nuh-uh, we don't move across the line like this. Instead, you got to reposition yourself. You're going to look a little bit like this. And when you're like that, then I'll move you across the membrane." So it kind of like grabs it and rotates it and then moves it across. So much of the pore is lined with hydrophobic groups that do not interact with water, which facilitates that rapid transport, because we don't want that water to be very strongly attracted. Instead, we want it kind of to be repelled. So we want hydrophobic residues. However, at the tightest point, each water molecule is separated from its associated water. So one water molecule is separated from 2 or 3 others. Now this is due to interactions with arginine and asparagine. So you've got lots of hydrophobic residues, but then you do have some polar positive residues and a polar uncharged residue. Polar positive being arginine, a polar uncharged being asparagine. Now, if this did not occur, rather than water, protons would simply jump through the channel. And we don't want that, because if protons are moving across, then we're going to have a pH change on one side. Next up are ion channels. And the ion channels that we'll talk about in just a little while are the sodium and potassium pumps. So ion channels are specific for specific ions, and they are going to move certain ions. They're going to move them at a very fast rate. I'm going to kind of breeze through this because we're going to talk about a sodium-potassium channel in just a moment. A potassium channel is 10,000 times more permeable for potassium than sodium, and potassium channels can maintain a diffusion limited rate of 10 to the 8th ions per second. So it's moving a lot at a very quick rate. Now, entrance to the internal pore is lined with 4 anionic side chains. It's going to attract cations. Another type of channel is a ligand-gated channel. So I mentioned earlier there are things that had gates, so basically barriers. Nicotinic acetylcholine receptor, or NAR, is found at the postsynaptic cleft. Binding of acetylcholine is going to open, allowing sodium to pass through. And so what's going to happen here is this protein complex looks a little bit like this. So a series of alpha helices. Now, those alpha helices are basically not going to let sodium through until they bind acetylcholine molecules, and they're going to undergo this rotation. And whenever they undergo that rotation, then this space right here in the middle gets wider, so more ions can pass through. And they're going to stay open, or this channel is going to stay open until this ligand, this acetylcholine, is removed. Now, there's many neurotoxins and chemical agents that affect either the binding of acetylcholine or act as an acetylcholine or acetylcholinease inhibitor. So basically that acetylcholine gets bound, opens that channel. Then acetylcholinesterase comes along and cuts it and breaks it into acetate and choline. Well, if that never happens, then that channel remains open. So some neurotoxins are going to inhibit this and block it from ever basically closing up again. There are also maybe a little bit more familiar or maybe a little bit more direct GLUT transporters. GLUT transporters are proteins that move glucose from outside of the cell to the inside of the cell. So basically they move it from the bloodstream into a cell. Now, what they're going to do, oh boy, is they are going to undergo a conformational change. So effectively, I always kind of think about it like this. My hands make up that glucose transporter. And whenever my hands come in contact with glucose, they're going to bind glucose. And for this, I'm going to use my wallet here as glucose. So they're going to bind glucose. And whenever they bind glucose, then they're going to open up so that glucose can be dropped to the other side. So glucose just came from the bloodstream. Now the protein undergoes this conformational change. Glucose is dropped to the other side of the membrane. GLUT 1 through 4, these are transporters. They are highly specific for glucose. GLUT 5 is highly specific for fructose. This allows glucose to bind on one side and be deposited on the other. Now, active transport is a transport mechanism that requires the input of energy. Now, some transport must occur such that solutes flow against their thermodynamic potential. In order for that to happen, energy has to be input. And an energy source and transport machinery are then coupled together. So we've got this broken down into two different systems. And these different systems are primary active transport and secondary active transport. What I'll do is I'll focus on primary active transport first. And here we have a simple model where ATP is hydrolyzed to make ADP and PI. So we're basically breaking that bond, we're hydrolyzing that ATP so that we can move X here from the outside to the inside of the cell. X is moving against its concentration gradient, as is evidenced by the single X inside the cell and the 10 or 11X on the outside of the cell. Okay, primary active transport requires ATP, it's active transport, and it's got one protein and it's moving one molecule. Okay, secondary active transport. You could look at this as what we are doing is we are aiming to move X right here into the cell. Now, how are we going to do that? Well, we're going to do this in a couple of different ways. We're first and foremost going to have to use two different proteins. So here's one, here's the other. Now, this protein on the left-hand side, I'm going to focus in on that. That protein is going to hydrolyze ATP and to make ADP and PI, just like what we saw previously. This is going to move X from inside the cell to outside the cell. So it's moving against its concentration gradient. And if I misspoke a moment ago, when talking about primary active transport, X is moving against this gradient. There's one here, and it's moving out where there's tons. If I misspoke, I apologize. So X here in secondary active transport is moving against this gradient. It's moving from a low concentration to a high concentration. Okay, so basically this right here is that first protein, that is our active transporter. We're using ATP. But then in conjunction with this, we are moving S, another metabolite, from an area of low concentration to high concentration. So S is moving in the opposite direction. So S actually, it has an energetic demand to get it from low to high concentration. And so then what we're doing, let me rephrase, I'll wrap it up and then I'll rephrase it. What we're doing is we're going to use a CHEM porter. So it's transporting two molecules, S and X, in the same direction. They're both going into the cell. This gets X into the cell, and it has no problem getting into the cell because it's moving from an area of high concentration to an area of low concentration. Okay, so that's the fate of X. And then, we can transport X out again using our ATP hydrolyzing protein right here. S, by comparison, it's basically only able to use X as kind of as a driver almost. It's almost like S says, okay, I need to get across the membrane. X says, well, I can make it across the membrane fairly easily. Ride with me. And so if X was absent, then this protein, let me put it in two ways. Then if X was absent, so we removed all of this, then the only way to get S across the membrane is to use a primary active transport protein, or it simply wouldn't move across the membrane. So let me rephrase all of this. X can freely move from one side of the membrane, or easily move from one side of the membrane to the other side of the membrane due to its chemical potential difference. S, by comparison, has a little bit of a difficulty because it's moving from low to high concentration. And so there has to be some motivating force to get S to the other side of the membrane. And it happens that X is able to do it and drive it across. Okay, now what I'd like to talk about is a sodium-potassium channel. Sodium-potassium channel is just that. It's moving 3 sodium and 2 potassium. Now, what it's going to do is it's going to move 3 sodium from the inside of a cell to the outside. So that's why we see 3 sodium in to 3 sodium out. So sodium is going from the inside to the outside. Potassium is coming from the outside of a cell to the inside of the cell. How are we doing this? Well, we're using water to hydrolyze ATP, and we're going to come out with ADP and PI. Now, this type of protein maintains intracellular sodium concentrations at a low level and potassium at a high level. It's crucial for all organs, but especially for neural tissue and the brain. ATP hydrolysis drives sodium out and potassium in. And this is, since they're moving 2 different metabolites in opposite directions, this is an antiporter. 3 positive charges exit for every 2 that enter. All cells expend a large fraction of their ATP to maintain the required cytosolic Na+ and K+ concentrations. Now here's a different diagram of what's happening. And this is a slide that you definitely want to pay a lot of attention to. So when it comes to our enzyme is going to be either E1 or E2. So E1 versus E2, that's a protein that's undergone a conformational change. And this is similar to what I was giving you the figure for our glucose transporter. But here what it is the protein E1 is open and it binds 3 sodium. Then it's going to undergo a change and it's going to release 3 sodium. But now on the other side, when it's released those 3 sodium, it's going to have something else to pick up. And that something else happens to be my stapler or these 2 potassium ions. I got to shimmy my stapler up, and then it's going to undergo a conformational change where it releases the potassium on the other side of the membrane. So those conformational changes are taking place with this protein. Now, it's a little bit complicated though, because if you look at this figure, there's a lot going on. And there's words mixed with letters, mixed with acronyms, mixed with colors. There's a lot going on. So I'm going to bring up the text, and then I'm going to kind of break down the different parts of it. So the additional text is that, or the key to this is that This sodium-potassium ATPase is the phosphorylation of a specific aspartic acid residue on the transport protein. So there is a phosphorylation event that takes place, and we'll get to that in just a moment, that initiates a change in the protein. ATP phosphorylates the transporter only in the presence of sodium ions. Then the aspartyl phosphate residue is the subject to hydrolysis only in the presence of potassium. Okay, so here's where we are. I'm going to start out in the upper left-hand corner, E1. Now E1 is able to bind 3 sodium. Where are we? We are on the inside, and that's what we want to do. We want to get sodium back into the bloodstream so it can, either be released, it can be, sloughed off, or it can go to where it needs to go. Okay, so what we get is our E1 that has bound to 3 sodium ions. Now, as soon as we have that complex, well then ATP, which ATP is stabilized by the presence of magnesium ions, that ATP will bind to our E1, giving us this E1 ATP 3 sodium ions. We're going to transfer a phosphate group from ATP onto E1, giving us this complex right here. This phosphorylated E1 3 sodium ion complex can then close off and open up on the other side of the membrane. So we're not talking about something going from here to there to there to there. Instead, my hands would be embedded in the lipid bilayer, open up, receive the sodium, receive the ATP, phosphorylate, open up. So this is E2, this is E1. Okay, so then what we've got is when we undergo this conformational change, our 3 sodium ions are released to the outside of the cell, so they're in the bloodstream now. That E2 is phosphorylated. That phosphorylated E2 is going to then have a high affinity for sodium ions, or potassium ions, sorry. Potassium ions from the outside of the cell. Those potassium ions are going to go to our phosphorylated E2. E2 is going to get hydrolyzed, releasing our inorganic phosphate. Now we have E2 that's no longer phosphorylated, and so it doesn't want to be in this shape, but instead wants to be in this shape, because this is our non-phosphorylated form of our protein. So it's bound to potassium, it's lost the phosphate, so it's going to go like this. Now all of a sudden these potassium ions that were kind of bound to our protein, they are released to the interior of the cell. Okay, so one thing that I want to add to this that I mentioned earlier, but I really want to emphasize, is the amino acid that gets phosphorylated. This is basically your kind of boilerplate standard look of an aspartic acid. But here we get a phosphate group added. So aspartic acid is one of the amino acids that we don't typically talk about as being phosphorylated. Typically when we talk about phosphorylation, we talk about a serine or a tyrosine or a histidine. But aspartic acid is the residue that's getting phosphorylated here. So E1, the form of that protein, and E2, the difference is what are they? E1 has a low affinity for potassium, so it releases it, but a high affinity for sodium. Then E2 has a low affinity for sodium, but a high affinity for potassium. Now, the way that some of these things ultimately all come together would be something like this. We have secondary active transport of the bloodstream and the intestinal lumen. Okay, so I'm going to go eat a piece of bread, and in that bread there's some salt. There's some sodium ions. There's also some glucose. Now those sodium ions and glucose molecules are going to be transported by a sodium glucose transporter after it's in my gut. Now those sodium ions are going to be packed up with another sodium ion, and they're going to go to that sodium potassium transporter that I just talked about. And they are going to move in the opposite direction as potassium ions, and those sodium ions are going to end up in my bloodstream. Now, that glucose that with the help of this symporter has made its way into the epithelial cell, that's going to be subject to a GLUT transporter, which is going to transfer that glucose into my bloodstream. That glucose can then circulate throughout my body, end up at a particular cell where glucose is going to, where GLUT2 is going to bind it, and then then in another cell down the line, another part of my body will be able to bind that glucose, or a GLUT transporter will bind that glucose, integrate it into that cell, and that glucose can then be metabolized and broken down into form a bunch of ATP. All right, well, that'll about wrap it up. I hope this was helpful.